Welcome to this video. We're going to go over a few things from chapter 14. So first, this starts with eukaryotic transcription. And there are several transcription factors involved. And you don't have to know their different names. We have here transcription factor 2D. That's what the TF stands for. It's going to bind to what's called the TATA box, or the TATA box if you prefer. We find these in the promoter region of almost all genes. So one transcription factor binds, and you can see the second transcription factor binds, and a third one that allows the RNA polymerase to bind. And then some other factors, they form what's called the pre-initiation complex or mediator, which binds and then allows this open complex to form. And that's going to allow the RNA polymerase to move to the elongation stage where it's adding nucleotides and making RNA. So here's the steps of that. Transcription factor 2D binds to the TATA box. Transcription factor 2B binds to 2D. Then RNA polymerase 2 binds to the core promoter. Transcription factor 2F is then bound to the RNA polymerase 2. And then transcription factor 2H and 2E are going to bind to the RNA polymerase 2. The transcription factor 2H acts as a helicase, so that's going to open the DNA and form what's called the open complex. And then transcription can start. RNA polymerase 2 will move down the RNA, or down the DNA, sorry. There are three polymerases that eukaryotes use that you should know. RNA polymerase 1 transcribes all the rRNA genes. So rRNA stands for ribosomal RNA, which are important for forming the ribosome, which we know is necessary for protein synthesis. RNA polymerase 2 does most of the work. It does all the protein encoding genes, also the snRNA, which stands for small nuclear RNA, and also non-coding RNAs, which we're not totally sure of their functions. And then we have RNA polymerase 3, which transcribes all the tRNA, which stands for transfer RNA. Transfer RNA is the one that brings amino acids to the ribosome. It also makes the 5S rRNA gene, so another piece of the ribosome. So you should know RNA polymerase 1 is the majority of the rRNA genes forming the ribosome. RNA polymerase 2 is all protein encoding genes, so that includes mRNA, which stands for messenger RNA. Then RNA polymerase 3 makes all the transfer RNA genes and the 5S rRNA gene. Another thing you should know is this protein called mediator that's found in eukaryotes but not prokaryotes. So it's basically RNA polymerase plus the 5 general transcription factors that we saw earlier that help form that open complex. And you don't have to know every piece of it, just know it's RNA polymerase 2 and the 5 general transcription factors. And it helps to switch between initiation and elongation of transcription. Then eukaryotic termination is different as well. In eukaryotes, we put what's called a poly-A tail on the end of every mRNA. That helps us know when to stop transcription. So there's two models, the allosteric model and the torpedo model. So there's no Rho protein like we had in prokaryotes. So this figure shows RNA polymerase goes along, and then it comes to this yellow region, which is the polyadenylation signal telling it to stop transcription. With the allosteric model, there are other proteins that are not shown that bind, and that causes the RNA polymerase to basically fall off the DNA, and that stops transcription. With the torpedo model, we have this, which I think is much cooler, we have this exonuclease protein, which binds to the RNA and starts chopping it up in pieces. And when that exonuclease protein catches up to the RNA polymerase, it causes it to dissociate from the DNA and fall off. So there's your allosteric model and torpedo model. So this question asks, which are features of transcription in eukaryotes only? So that would be the presence of 3 types of RNA polymerases, mediator controlling the switch to the elongation phase, addition of the 7-methylguanosine cap, which we'll talk about in a second. The promoter consists of a -35 and a -10 sequence. That would be the same in eukaryotes and prokaryotes. And then termination occurs according to the allosteric or torpedo model. That would apply to eukaryotes only. So after the mRNA is made in eukaryotes, there is some processing that happens. One thing is that it's spliced, and this allows us to make several different proteins from the same mRNA depending on how we splice it. It's also capped at the 5' end. That cap is going to help it know where to go, because in the eukaryotes, the mRNA has to leave the nucleus and go out and find a ribosome, and that's one of the jobs of that cap at the 5' end. And then we add what's called a poly A tail at the 3' end. And that poly A tail is going to control basically how long this mRNA transcript lasts. Every time it's transcribed, some of those in the poly A's are just extra adenines at the end. Every time it's transcribed, some of those adenines are going to be lost, and once enough of them are lost, it can't be transcribed anymore. So the 7-methylguanosine cap of eukaryotic mRNA serves which of the following functions? And it is recognition of the 5' end of the transcript by the translation machinery. That way it knows how to enter the ribosome. So next thing we're going to talk about is splicing. So a lot of mRNA is what we call introns, which is not protein coding regions. And so after it's transcribed, we have to get rid of that. This shows a 5' splice site, a branch site in the middle, and a 3' splice site. And some proteins are going to bind at the 5' splice site and then at the branch site. And they form what's called the spliceosome, along with some other proteins that bind at the 3' splice site. One of the first steps, the 5' splice site is going to be cut. These proteins make the intron form a loop and brings those splice sites closer together. Then the 3' splice site is going to be cut, and these 2 exons are going to be connected, and then that intron is lost and it's just degraded. We don't need it anymore. Last thing I want to point out is this table showing the difference between transcription basically in bacteria and eukaryotes. So the promoter is pretty similar. In eukaryotes, we have this TATA box. For RNA polymerase, just 1 in bacteria, 3 in eukaryotes. For initiation in bacteria, we need sigma factor. In eukaryotes, there's 5 general transcription factors. Elongation in bacteria requires the release of sigma factor. In eukaryotes, it requires mediator. Termination in bacteria is either row dependent or row independent. In eukaryotes, it uses the allosteric or the torpedo model. Splicing in bacteria is very rare, and there is some self-splicing. In eukaryotes, it's very common, and it uses the spliceosome proteins. Capping does not occur in bacteria and does in eukaryotes because bacteria doesn't have a nucleus. Tailing happens in bacteria, but it's a little different. It actually promotes degradation in bacteria, whereas in eukaryotes it promotes stability. RNA editing does not occur in bacteria, and it occurs occasionally in eukaryotes. If you have any questions, please email me.